Have you ever thought about why water is so important for life? When we look for life on other planets, often what we're looking for first is liquid water. Water is a very simple molecule, right? As we know, it just has an oxygen and, of course, our two hydrogens to make up the famous H2O formula. So it's a very small and simple molecule. But as we'll see throughout biochemistry, it's critically important for our living systems. If we look at a variety of different biological organisms or individual cells, what we see is that water is a huge component of all of these different organisms, right? So here we're looking at the different molecular components of an E. coli cell. You can see water makes up 70% by weight of all of the total components of our cells. Well, this is bacterial cells in this case. All of the other biomolecules, which we'll be focusing on a lot throughout this course, make up overall much smaller percentages of the total content of the cell. Even in scripture, water takes a very special role as a symbol of life, and we see that the Bible often refers to living water and this important role of water as a symbol or a source of life. So in John 7, it says, "Whoever believes in me," as scripture has said, "rivers of living water will flow from within them." Again, emphasizing that special role of water as a molecule of life, essentially. So why is this? What actually makes water so special, right? Why do we look for it on other planets as a sign of life? Why do we see it as such a major component of our body? And why is it a symbol of life in the body or in the Bible? So to answer this, we need to, of course, go back to the basics and look at what our water actually is in terms of its atomic structure and how it behaves. So as we look at the periodic table, of course, one of the things that we usually learn from the periodic table is our electronegativity trends as one of our key periodic trends. So you have oxygen over here on the far right. We have hydrogen over here on the far left. Remember that electronegativity increases as we go from left to right and from the bottom to the top of our periodic table. So oxygen, of course, is much more electronegative compared to our hydrogen. So now as we go back to that oxygen water molecule structure, of course, we know that our hydrogens are going to have a partial positive charge as the electron density is pulled towards our oxygen. And of course, our oxygen gets a partial negative charge, which of course ends up kind of focused on these lone pairs that we have on our oxygen. So we have an uneven distribution of that electron density and that charge within our water molecule, which then, of course, influences how our water molecule behaves. Now, one of the key things that this does for our water molecules, it allows us to make very strong hydrogen bonds, right? So as you should know from earlier courses, right, a hydrogen bond forms when we have a hydrogen, which is going to have that partial positive charge because of the attachment to an electronegative atom. So that's our oxygen with the partial negative charge. And then those partial positive and negative charges can interact with other nearby atoms, right? So we take a partial negative charge from a nearby electronegative atom that we call our hydrogen bond acceptor, whereas the hydrogen and the oxygen are our hydrogen bond donor, right? So then we can form a variety of hydrogen bonds as shown here, right? We can take electronegative oxygens and hydrogen bond them with our hydrogens, or we can take nitrogens as shown here. And of course, again, our hydrogen just has to be attached to some electronegative atom. In biochemistry, we commonly see it attached to oxygens and nitrogens. Now, hydrogen bonds are interesting because they're one of the strongest intermolecular interactions that we see in chemistry. So it's, again, mostly based on this difference in electron distribution, or the partial positive and negative charges. But it actually has some covalent nature as well. So it's roughly 10% covalent and 90% electrostatic. So it's actually a very strong interaction between our hydrogen and those nearby electronegative atoms. Now, one of the other key features of our hydrogen bond is that it's actually very dependent on shape. So as we have our electronegative atoms interacting with each other in this hydrogen bond, right, our partial negative and partial positive charges, our strongest interaction occurs when they're all lined up in a single line between our three atoms that are involved here, right? So our oxygen, our hydrogen, and the oxygen or other electronegative atom on the other side. If we have those so that they're not lined up, so over here we have our hydrogen in the middle, but then we have one of our electronegative oxygens directly above it, and the other one coming down to the side, right? So now instead of being a linear hydrogen bond, we have a bent hydrogen bond, and this actually ends up being significantly weaker. So through this, our hydrogen bonds will actually affect the shape of our molecules as well. And we'll look a little bit more at that later. So for our water molecules, they're going to be forming many hydrogen bonds through that electronegative oxygen and hydrogen within our water molecule, right? So the hydrogens on one water molecule will be, of course, acting as our hydrogen bond donors, and they'll be interacting and forming hydrogen bonds with the nearby oxygens of our water molecules. So we have our hydrogens, of course, that can both act as hydrogen bond donors, and then our oxygen, which can act as hydrogen bond acceptor through the partial negative charge in the lone pairs around our oxygens. So within our water, we expect to form then a network of hydrogen bonds between all of our many water molecules in any given collection of water. So if we have frozen water, we end up getting this perfect kind of hexagonal arrangement of our water molecules, as you can see here. So this is our crystalline ice water. We can see, ideally, each of our waters will form 4 hydrogen bonds. Each of our hydrogens, of course, making hydrogen bonds to nearby oxygens, and then each lone pair on our oxygens forming hydrogen bonds to nearby hydrogens. So you end up with this very precise arrangement of our water molecules in order to get these 4 possible hydrogen bonds. Now, of course, the 4 hydrogen bonds are only possible if we arrange perfectly in this sort of crystalline structure. When we have a more dynamic liquid state, we end up forming less hydrogen bonds, so only 3.4 on average. That's just because our water molecules are moving around more, and so they don't want to stay in this rigid structure. So this actually leads to one of the very unique properties of water. So in the solid state, when we form this crystalline structure with our 4 hydrogen bonds for each water molecule, we actually end up being less dense than in the liquid state. In the liquid state, our water molecules are able to pack a little bit more closely together because they're not so concerned about having this perfect arrangement in order to get their 4 hydrogen bonds, right? So they're able to kind of move around a lot and still get our 3 or 3 to 4 hydrogen bonds as they're kind of moving around that liquid state, the 3.4 on average. But they're not as carefully positioned as here in our ice crystals. So you can notice, of course, these kind of empty spaces here that result as our water molecules sit in this perfect structure with their 4 hydrogen bonds that then lead to that lower density of water. Now, from a biological standpoint, the consequence of this is that we end up with our ice floating on top of bodies of water. And so things like ponds and lakes don't completely freeze over during the winter to allow fish and things like that to stay alive at the bottom of the pond. So our hydrogen bonds are very common inside of our water, right? Now, what significance does this have for biochemistry, right? So far I've talked about biology, right, of the hydrogen bonds within our water molecules causing ice to float and then allowing fish to survive. How does this connect to our biochemistry? So it turns out it has a very, very huge connection to biochemistry. So Linus Pauling, who won the Nobel Prize in Chemistry back in the 1950s, he said that I believe that as the methods of structural chemistry are further applied to physiological problems, it will be found that the significance of the hydrogen bond for physiology is greater than that of any other single structural feature. So clearly, he thought that hydrogen bonds were extremely important for our biochemical systems. And I'll show you a few of the reasons why that hydrogen bonding in our water is so important here. So first off, let's look at some of the different types of hydrogen bonds that we see within our biological systems. So here, of course, we have our water molecules, which, as we know, like to form hydrogen bonds. And they can form hydrogen bonds to a variety of different functional groups that we commonly see in biological molecules, right? So we see alcohols that can hydrogen bond to water. We see carbonyl groups such as ketones that can hydrogen bond to water. And all of these are commonly found in our biochemical compounds. In addition to hydrogen bonding with our water molecules, we can also see a lot of hydrogen bonding inside of our biomolecules, right? So between peptides, which we'll talk about more later, we can have hydrogen bonds occurring between our oxygens and our carbonyls and our amines. We also see hydrogen bonds forming in DNA. So here are two of our common bases of DNA, thymine and adenine. These make up the T and the A that you might have seen in genetics classes or basic biology courses. And we see that these guys are also using hydrogen bonds to pair up between these bases. Now, of course, again, as we mentioned earlier, right, one of the other cool things about a hydrogen bond is that it does like to have that specific linear orientation in order to form the strongest hydrogen bond. So not only do we see these hydrogen bond interactions creating relatively strong connections between various biomolecules and each other, as well as biomolecules in water, but we also see it kind of forcing our biomolecules into specific orientations in order to form the perfect, strongest linear hydrogen bonds, right? So if I was to take my adenine and kind of bend it over here to the side, imagine just taking this and maybe turning it like 30 degrees to the side. That would put my two hydrogen bonds here in a bent shape instead of a linear shape, and would make a weaker interaction, right? So we have a preference for this linear shape of my two interacting DNA bases here. So our first key effect of water in our biological systems is that it can form a lot of hydrogen bonds with our biomolecules. And again, that affects our shape based on the strength of the linear hydrogen bond and the weakness of our bent hydrogen bonds. Now, another key factor for the importance of water is its use as a solvent. So water is a very good solvent for charged and polar substances, which in the case of our biomolecules, this includes the amino acids that make up our key proteins. This includes alcohols, as I mentioned before, and it also includes our carbohydrates. Carbohydrates generally contain a lot of alcohol functional groups, which again, we'll talk about that more later this semester. It's not a great solvent for more nonpolar substances. So this can include nonpolar gases, various aromatic compounds, and of course, any kind of hydrocarbon or aliphatic chains. So most of our biological molecules are going to fall into these cases up here, are more charged and polar substances. And so this will allow it to mix in our body, which of course is largely made up of water, as I mentioned earlier. We will still see some of these nonpolar substances as well. And actually, the fact that water is not a good solvent for those nonpolar substances will affect how they behave within our biochemical systems. So as we look at some examples of different biological molecules, so we see our polar groups are marked in blue. These are going to easily mix with our water and form those strong hydrogen bonds. Our nonpolar groups are marked here in yellow. So you do see we've got some large nonpolar components on some of our biological molecules. These are mostly tend to be our hydrocarbon-based functional groups, right? Notice our alkanes here, carbon-carbon single bonds, alkenes, those carbon-carbon double bonds. We have aromatic rings, etc., all creating these very nonpolar groups. So what actually happens when we dissolve those various biological molecules in our water-based environment in biochemistry? So of course, in general chemistry, I mean, we talk a lot about the dissolution process, right? We see how with charged or ionic compounds, right, our water molecules use those partial charges, right? Remember, we have the partial negative and positive charges on our water molecules, and we use that to interact with our negative and positive charged ions, kind of pull them out of the crystal, and then they end up surrounding them with these kind of, we call it a solvation shell of water molecules surrounding our individual ions. So we get a shell of water molecules with those positive hydrogens pointing in around our negatively charged ions, and we get a shell of water molecules with our negative oxygens pointing in around our positively charged cations. So of course, this kind of simplistic picture you've likely seen before. Again, in our general chemistry, we talk about this. But in biochemistry, we want to take a little bit of a closer look here and see what's going on from an energetic perspective. So one of the key equations that we need to think about whenever we're looking at the energetics of a chemical process is our Gibbs free energy equation. Right? So Gibbs free energy is our delta G over here. Remember, if this is negative, that means that we have a spontaneous process. And if it's positive, then we are non-spontaneous, right? So if we want a process to occur, we usually need to have a negative delta G. If it's positive delta G, then we have to add in extra energy in order to try to make that happen. So our Gibbs free energy depends on our delta H, our enthalpy, which this is based on kind of what interactions we're forming or what interactions we're breaking. So our interactions formed or broken. And it also depends on our entropy over here, our delta S, which refers to kind of the overall order of our system, right? So if you have more entropy, that's a more disordered system. If you have less entropy, that's a more ordered system. You can notice in terms of our signs here, so we have a negative sign in the middle. So if we have a positive change in entropy, right, we're increasing the amount of disorder in our system, that will lead to a negative term over here on the right, which will then contribute to a more spontaneous process overall. For our interactions, if we're breaking interactions that cost energy, it's going to be a positive delta H. And if we're forming interactions, it's going to be a negative delta H contributing to a more spontaneous process. So our overall spontaneity of any given process is going to be governed by this balance between the interactions that we're forming, the interactions that we're breaking, as well as the change in the entropy or the order of our system. So in the case of dissolving our salts, so this is a favorable process, right? When you pour salt into water, it dissolves without you having to do anything, right? You don't have to heat it up. You can just leave it and the salt will dissolve. Now, heating can help speed up that process potentially, but you don't have to heat it in order for that salt to dissolve. So it's obviously a spontaneous process. Our delta G is negative. Now, what about our individual terms here? Our enthalpy, the delta H, and our entropy, the delta S. So as we're adding that salt into our water, we are going to be forming favorable electrostatic interactions with our solvated ions, right? So as we're forming interactions, that's a negative delta H, although you do have to realize that there's a positive delta H here also from breaking the interactions within our original salt crystal. However, as we are breaking those ions out of our ionic compound, we're also increasing our entropy, right? So within our salt crystal, they're all closely packed together in this very rigid structure, right? It's a very ordered salt crystal over here. And when we break those ions out, they become much more free, right? And that corresponds to an increase in entropy, right? Our individual ions can move around. There's a lot more disorder compared to this very rigid and organized salt crystal. So our entropy increases. That's a positive delta S, which of course, due to the negative sign, leads to a more negative delta G, or a more spontaneous process. And finally, the last key thing that's happening here is that the water itself is actually weakening the ionic interactions between our negative and positively charged ions. So the attraction between our ions is actually based on this equation shown here down at the bottom. F is the force of attraction between those ions. Q here is the charge of our individual ions. R is the distance. So of course, as we separate ions, we can also reduce the strength of that interaction between them. And our little epsilon Greek symbol here refers to this dielectric constant. Now, the dielectric constant is related to the polarity of our solvent, right? So it's essentially looking at how much of a charge distribution there is within our specific solvent. Now, water, because of, again, that charge separation between our electronegative oxygen and our low electronegative hydrogen, has a very high dielectric constant, right? It's essentially shielding our ions from feeling that force as it's kind of blocking it through the small charges of our own water molecules. So as it starts to pull our salt ions away from the crystal structure of the ionic compound, it actually reduces this force as they get shielded through that high dielectric constant. So these three key factors all contribute to a negative delta G in a very spontaneous process for our water dissolving salts. You could follow a similar reasoning for other polar compounds. So of course there we're again forming favorable interactions, although of course it's a little bit simpler there because we don't have the ionic charge and the shielding effect to account for in those scenarios. Now what happens if we put a nonpolar solute into water, right? So as we saw earlier, there are various biomolecules that have nonpolar components in addition to those polar components. So how do those mix in our water-based systems? So when we place those nonpolar compounds into our water environment, we see this thing that we call the hydrophobic effect. This is actually a very important factor in biochemistry. It affects our proteins and how they fold, how they form their specific structures in the body. It affects interactions between those biomolecules, particularly our proteins, leads to the formation of things like lipid micelles and various other binding events within our biochemical processes. So what is this hydrophobic effect? Well, to understand this, we have to look at what an individual nonpolar molecule looks like when it's in our water solvent. So our water, of course, normally it likes to hydrogen bond with other water molecules. And so we end up with what we call these sort of kind of flickering clusters of water molecules, right? Where they're all kind of swinging around trying to find other water molecules to hydrogen bond with, right? So you end up with hydrogen bonds forming, but then our water molecules can keep moving around. So we'll break one hydrogen bond and turn and find another water molecule that it can then hydrogen bond to, right? So right now, these two that I've circled in red are bonded together. But if we just turn this one down here a little bit, we can hydrogen bond with this water molecule down here instead, right? So normally our water in solution is very free, right? No matter what it does, it can always rotate and see another water molecule that it can hydrogen bond to, right? Again, like if we look at this water molecule on the top right, currently it's bonded to these two water molecules over here, but we can easily rotate it and hydrogen bond with this water molecule on the top right as well, right? So there's always water surrounding it, and so there's always possibilities for hydrogen bonding, right? This means that our water molecules normally have very high entropy, right? No matter what they do, they can keep turning and forming hydrogen bonds with new water molecules. Now, if we add a nonpolar solute, so that's represented right here in the middle. So this is a fatty acid where we have a carboxylic acid at one end, which is hydrophilic. It can interact with water and form those hydrogen bonds. But then we have this long hydrocarbon chain, which of course is nonpolar or hydrophobic and doesn't interact through hydrogen bonding with our water molecules. So when we have this nonpolar solute here, we can't form hydrogen bonds between our water molecules and our carbon chain here. So now if we look at all these water molecules right here along the surface of our nonpolar solute, every time they look towards our carbon chain, they can't form hydrogen bonds there. They can only form hydrogen bonds pointing out away from our nonpolar solute. So essentially, these water molecules end up being restricted by our nonpolar solute, right? They can no longer freely turn around as much as they want because they can really only form hydrogen bonds in one specific direction, right? Away from our nonpolar solute. In solution, again, remember they could turn around whichever direction they want, and they're always going to see another water molecule that they can hydrogen bond to. The water molecules at the surface are restricted, right? They can only form hydrogen bonds pointing away from our nonpolar compound. And so they end up being restricted and only wanting to kind of look out in one direction to get their hydrogen bonds. So it's not necessarily changing the hydrogen bond formation, right? Our waters are still forming hydrogen bonds. It's just kind of restricting where they can get those hydrogen bonds, right? Anytime you hear restriction, that should tell you, right, that it's more ordered, right? It's held within a specific arrangement, a specific orientation. And so we've increased the order and therefore decreased the entropy. So when we add a nonpolar solute, we're going to end up creating lower entropy for the water molecules that surround our nonpolar solute. And that's compared to the high entropy that we normally see for our water molecules in solution. So if we put that nonpolar solute into our water system, into our water solvent, right, we form this cage of highly ordered water molecules around our nonpolar solute. That creates low entropy, as I just said. And of course, lowering our entropy, that gives us a negative delta S term. That becomes positive when we add this negative sign in front. And so we create a positive delta G. So it's unfavorable for our nonpolar solute to dissolve in water. Now, of course, you already knew this, right? I told you earlier that nonpolar, of course, means that it doesn't dissolve very well in water. How does this relate to our hydrophobic effect? Well, what happens if we add multiple nonpolar solutes into water together? So here we have 3 different fatty acids, which I'm circling here, right? That yellow portion, again, is our nonpolar portion. And for each of these, you can notice this light blue region of our ordered water molecules surrounding those nonpolar solutes. So right now we have a lot of water molecules surrounding each of these, creating a large decrease in our entropy. Well, what happens if instead of spreading out these different lipids, these different fatty acids, we combine them all together, right? So we still have our same 3 nonpolar molecules, but now we've placed them next to each other. And you'll notice that now we only have a few water molecules around these 3 nonpolar molecules compared to the large number of water molecules that we had before. Right? If you look at the number of water molecules represented here, right, somewhere around 13 or so, about a dozen water molecules. In the previous scenario, right, we have around 10 or so water molecules around each of our nonpolar solutes, or around 30 molecules overall. Right? So there's a lot more ordered water here than there is in this case when we cluster our 3 nonpolar solutes together. So if we have less of our water getting ordered, then our entropy is going to increase, right? Less order equals more entropy. So if we take the nonpolar portions of our molecules and aggregate them or cluster them together, we can actually increase our entropy by reducing the number of water molecules that are ordered. So our nonpolar solutes like to cluster together, and we call this the hydrophobic effect. If we have a large amount of nonpolar solutes, we can start to form large and complex structures like our micelles. So in a micelle, you actually have enough nonpolar solutes that you can cluster all of the nonpolar solutes together in the center of this micelle, and then our hydrophilic portions can be on the outside interacting with our water, and we essentially remove all of those ordered water molecules, right? There's no water now surrounding our nonpolar solutes, and so we've been able to increase our entropy by clustering those nonpolar solutes together. So our entropy, again, is increased as we're eliminating that ordered shell of water molecules. So this is one of the fascinating aspects of biochemistry, is that we can actually create something that looks relatively ordered by increasing our entropy, right? So our micelle looks relatively organized, right? We have all of our nonpolar molecules kind of packed together in this nice little spherical structure with them all organized on the inside. But overall, we've actually increased the disorder because we've allowed much more freedom for our water molecules themselves. So I think it's a pretty interesting testament to God's amazing design here, right? He creates it such that even in something that we perceive as being random, he's still able to create a sense of order, even as our water molecules are still disordered here and we have a favorable process, right? So we're able to create a complex structure while actually increasing our entropy and not get in the way of those thermodynamic problems of decreasing our entropy as we increase that order here. So the hydrophobic effect is going to allow our nonpolar solutes to cluster together and then still be contained within our water environment, even though, of course, they don't like to dissolve in water. Now, our hydrophobic effect doesn't just involve whether something can dissolve or not in our water-based solvents. It can also affect interactions between our biomolecules. So in many cases, our molecules can actually use hydrophobic portions or nonpolar portions in order to interact with other molecules. So in this case, we have a substrate molecule that wants to bind to an enzyme. And if our substrate is somewhat hydrophobic, we'll have, again, a layer of ordered water molecules around that particular substrate. If we have a hydrophobic or nonpolar site in our enzyme, we'll also have ordered water molecules around that enzyme. Now, as we bring our substrate closer to that enzyme, we're going to be able to kick out all of these ordered water molecules as we form that hydrophobic interaction between our two biomolecules here. Right, so we can actually, again, increase entropy by kicking out our water and making it more disordered, and increase the favorability of this interaction. So our enzyme substrate interaction then gets stabilized through both that hydrophobic effect, but then we can also, of course, use other interactions such as our hydrogen bonding, all those typical kind of intermolecular interactions to help strengthen this interaction as well. Now, everything that we've looked at so far has been focused on how do different solutes dissolve in water, right? And what effect that has in the structure of our water. Now, of course, as we're dissolving our solute in water, right? So we can look at it from the solute perspective of whether it's going to mix well with our water. But we can also think about how that solute is going to change our water itself. Right, so in this case, we're looking at a small alcohol. So this is ethanol over here on the left. And ethanol can mix very well with water because of the hydrogen bonds that can form between our alcohol and our water molecules. Right, so you can see all of these dashed blue lines are representing those hydrogen bonds. And since it can form lots of hydrogen bonds with our water, it can mix very well with our water molecules. But even with something like ethanol, right, there are portions of ethanol that can't hydrogen bond with our water molecules. Right, so these carbons over here still can't hydrogen bond. So it is going to have some effect on our water. right? Waters that are near those carbons aren't going to have as many possibilities for hydrogen bonds as our other water molecules. So this leads to typical effects that we've, of course, heard about before in general chemistry, right? So we get freezing point depression. So as we add solutes into our water, it disrupts the hydrogen bonding of our water and lowers our freezing point, right? So it becomes easier to break apart that crystal structure of water because, of course, some of our water molecules are going to be interacting with our solute, and they're not going to be able to form that perfect rigid structure that we looked at earlier in our ice crystal. Of course, at the same time we have this freezing point depression, we also get our boiling point elevation, again, just based on the slight difference within our water structure that's created by the addition of these solutes. Now, one very important effect of solutes on water in our biochemical systems is osmotic pressure, right? So since those solutes are interacting with our water molecules and kind of disrupting the normal arrangement of our water molecules, this actually creates this osmotic pressure where our water wants to move from areas of high water concentration relative to our solute, so low solute concentration, to areas of low water concentration or high solute concentration, right? So our water kind of wants to move around in order to equalize the overall solute concentration, right? So this is just based on our water molecules kind of trying to move to where they can get more hydrogen bonding compared to those disruptions caused by our solute, right? So we're kind of evening out the amount of solute-based disruptions within our water. So this creates this osmotic pressure, right, where water is trying to move in order to even out that solute concentration and reduce kind of the relative effect of our solute. It's affected by the concentration of all of our solutes in solution. If we have dissociated compounds like our ionic compounds, each of those individual ions that break apart will affect our overall osmotic pressure, right? So if you remember back to the ionic compound, right, the anion would break off and you get water molecules surrounding that. Again, that represents a change in our water structure. The anion would break off separately and you get water molecules around that. Each of those would be affecting our overall water structure and then creating this osmotic pressure. So if we have two different solutions that are separated by a semi-permeable membrane, so we have in this case pure water, so that would be a very low solute concentration. And we have another solution here that has our solute dissolved. And we have a semi-permeable membrane that just blocks movement of solute between these two chambers. So our water is going to flow in and dilute that solute. And we're actually then pushing our water level higher based on that osmotic pressure of our waters wanting to move in here in order to even out that concentration gradient. So you can force this to stay in the same case. You could, if you use a piston to kind of force this to stay at the same concentration, you can actually use that then to measure the actual osmotic pressure, or even to force it to go in the reverse direction, which would be what we call reverse osmosis. Now, within our biochemical environment, this can have a huge effect on our cells within our body, right? So with a cell, right, you have a membrane that's kind of separating your solutes outside from your solutes inside, right? So we get separate pools of extracellular solutes and intracellular solutes that maybe can't pass through the membrane very easily. If we have too much solutes outside, our water will want to flow out and it will cause our cell to shrink. If we have too many solutes inside, our water will want to flow in to try to dilute those solutes, and you can actually end up breaking your cell. So this would be our hypotonic conditions versus our hypertonic conditions based on a difference in concentration inside or outside of our cells. So again, just based on how many solutes are dissolved in water and then how that affects our water structure, this then causes our water to move around to try to change that impact on our water structure, and can then lead to large-scale changes, even the disruption of our cells themselves. Okay, so this has been mostly just looking at water as a solvent. So that's another of the key roles that water has within our biochemistry. So of course, in organic chemistry, we look at different condensation or hydrolysis reactions where water can be used to form or break specific bonds in organic chemistry reactions. And of course, one of the most important examples of water-based reactions is actually acid-base reactions, right? Our water can either donate or accept hydrogens. Of course, our water ionizes to form extra protons within our aqueous-based systems, as well as hydroxide ions that can either then donate or accept hydrogens from our other biomolecules. So in our next lecture, we'll take a closer look at these reactions, specifically our ionization and acid-base reactions within our water-based environments.